Hello students, let's start. Master NEET Bio for You presents Biology 360. We are into series A2 where we are discussing about genetic numerical. I am Professor Sushil Mishra. Will give you some special tricks which will help you to solve numerical in a very quick way. Let's start with our next question. How many pure lines are obtained when a trihybrid, that is capital A small a, capital B small b, capital C small c, is crossed with a trihybrid capital A, capital B small b, capital C small c? Now remember, this question can also be asked as trihybrid is selfed. They want us to find how many pure lines are obtained. Now remember, for this I had got a very small and easy trick. Just see, it is given by a formula. Pure line is always equals to 1 by 2 raised to n. Now where n stand for number of heterozygous condition, remember they want us answer in percentage, so I have multiplied with 100. Now remember, it's a trihybrid, so 1 by 2 raised to 3, which is 1 by 8, 1 by 8 into 100, we get answer as, right, 12.5%. Let's proceed for the next question. Remember this formula, I told you number of pure line is always equals to 1 by 2 raised to n, where n stand for number of hybrid condition. You can use this formula for monohybrid, dihybrid, trihybrid and so on. Next question. In Hardy-Weinberg's population of peppered moth, remember Hardy-Weinberg, evolution topic you have learned. Again, numerical comes, please be very careful. Three types of genotypes are homozygous dominant, heterozygous and homozygous recessive. That is capital B, capital B, capital B, small b, small b, small b. If the frequency of dominant allele is 0.3, then what will be the percentage value of black-winged moth in the same population? Now remember, for Hardy-Weinberg, whenever they had given you Hardy-Weinberg, so we know P plus Q is 1. What is P and Q? P is the frequency of dominant allele, Q is the frequency of recessive allele. So we know P plus Q is always 1. Here in our case they had given the value for P. So I will substitute it and get the value of Q. Remember, this is your always a target to get the value of P and Q, that is frequency of dominant allele and the frequency of recessive allele. Now once you get it, now check what they want. They want us to find black-winged moth. That means they want us to find recessive individuals. Recessive individual frequency is given by the formula Q square into 100. Why 100? Because they want us to express it in percentage. Q was frequency for recessive allele. So Q square into 100, we get the value as 0.7 square into 100, that comes out to be 49%. That's right. Now remember, generally for such question they ask for heterozygous individual. Now remember, frequency for heterozygous individual is 2pq, not just pq, it is 2pq for heterozygous individual. Clear? That's great. Now let's move on to next problem. A trihybrid plant in which first character shows dominance, second shows incomplete dominance. Incomplete dominance, we know red and white cross, we get pink. So that is called as incomplete dominance. And the third shows codominance. Codominance is the case where both the characters are equally expressed. Right? In both incomplete dominance and codominance, our ratio is 1:2:1. Geno as well as phenotypic ratio. Yes. Now total there are 3840 plants are obtained in the progeny. How many of this will show dominant for first? Now remember, be careful, dominant for first, intermediate trait for second and both the trait for third. After reading question, most of the students quit it. Go one by one. What they are asking us, dominant for first. Now for first, how many dominant is there? Ratio check out for F2 generation 3 by 4. So I will take 3 by 4. Intermediate trait for second. Intermediate trait for second means pink color. What is the ratio? 2 by 4. 2 by 4 is 1 by 2. So into 1 by 2. And both the trait for third means again that cow example we have seen, roan example where red and white both are equally expressed, that again the ratio become 2 by 4. 2 by 4 means 1 by 2, so I multiplied with 1 by 2. So this is my proportionality which I get here. Now 3 by 4 is for dominant, 1 by 2 for incomplete dominant, and 1 by 2 for codominance, multiplied with total number of offspring gives me the right value for required problem. So that comes out to be 720. So I got my answer in just fraction of seconds. Let's proceed. Now once again I have taken example for pedigree analysis. Identify the type of given pedigree. Now look at the picture. I always say pedigree go by elimination method. So here, if you see the case 1, father is having the disease, the child are not having a disease. So definitely it's not a dominant. Reason, dominant is always expressed in next generation. So my first case, dominant X-linked feature goes away. Remember, if it was dominant, then definitely from father it will go to the daughter and daughter will show the feature. It is not showing, so it is not a dominant X-linked. So one goes out. Now second, look, father is not having disease, but daughter is having disease. This is not possible in case of X-linked recessive. If X-linked recessive, remember, if daughter has a disease, by default, the father should have a disease. Yes or no? Because daughter having disease, that means it has both defective gene. One should come from father, one should come from mother. So here, father is not having disease, but daughter is having disease. So recessive X-linked goes out. Next case. Here, if you look, female is having disease. If female is having disease, then it cannot be Y-linked, because Y-linked is always seen in case of male. So if 3 options are eliminated, by default my answer becomes cytoplasmic inheritance. Now remember, mitochondrial DNA or chloroplast DNA, they represent cytoplasmic inheritance. Cytoplasmic inheritance is always maternal inheritance. Let's go for next problem. The only son of Ravi's grandfather is hemophilic. The only son of Ravi's grandfather is hemophilic. If Ravi's father-in-law is also hemophilic, then what will be the probability of Ravi has a hemophilic son? Now let's analyze one by one statement. The only son of Ravi's grandfather is hemophilic. It means to say that Ravi's father is hemophilic. Grandfather's only son, that means Ravi's father. So if Ravi's father is hemophilic, they have not specified about Ravi's mother. So by default, if anything is not specified, we consider it as normal. So that means Ravi's mother is normal. So by default, Ravi is also normal. Now go for second case. It says that if Ravi's father-in-law is hemophilic, it is not talking about mother-in-law. So that means mother-in-law is normal, father-in-law is hemophilic. So Ravi's wife will be carrier. So Ravi's wife is carrier and Ravi is normal. So let's go for a cross between normal male and carrier female. A cross between normal male and a carrier female gives you 4 offsprings. First is normal daughter, second is carrier daughter, third is normal son, and the fourth is hemophilic son. Now what the question says that what will be the probability of Ravi has a hemophilic son? Remember, they are not asking what percentage of the son will be hemophilic. They are asking what is the probability of having hemophilic son. That means among all the offspring, what is the probability of the hemophilic son? Then it will be 1 by 4. Now 1 by 4, when I express in percentage, it comes out to be 25%. So this is clear. This way you should analyze the problem. Your problem will be solved in very quick manner.